we are discuss about the topic of palynology. Palynology is the study of spore and pollen that shape, symmetry, aperture, and etc. etc. So, what is palynology? Palynology is the study of pollen and spore wall. It has been recognized as a discipline that has made significant contributions to taxonomy. Palynology is much a part of embryology as seed coat anatomy, the branch of science with the study of fossil and living palynomorphs. On the basis of pollen morphology, all angiosperm families are divided into two groups: Uropalynous, Stenopalynous. The next one is Palynomorphous. Palynomorphous include microscopic plant and animal structures composed of pollen grains, setae, or related compounds that are highly resistant to most forms of decay other than oxidation. Palynomorphous are abundant in most sediments and sedimentary rocks and are resistant to the routine pollen extraction, produced including strong acid bases, acinolysis, and density separations. The palynomorphous size between 5 to 500 μe. And these are the common palynomorphous categories. It is divided into 13. We are discussed about the morphology of pollen, embryospike pores, and pollen. The next one is pollen morphology. Pollen morphology is conducted as an aid to the morphological study and a significant tool for modern taxonomists for the delimitation of species. Is studies using L, M, and SEM and is important to visualize the general features of pollen grains, including example, symmetry, and that pollen shape, size, aperture, number, and location, as well as ornamentations. And the next one is embryophyte spores. Embryophyte spores are microscopic unicellular reproductive cells of keratin vascular plants. These spores are extremely resistant and are easily transported by wind and water. They are useful biostratigraphic tools, particularly in freshwater environments. The source variable surface sculpture, ornamentation. Next one is the main thing of pollen grains. Pollen grains are the containers of the male gametophyte generation of seed plants. They are produced in the male organ of the flowers. Pollen producing is a strategy by which seed plants become free from dependence of standing water from fertilization. Pollen accrues by transferring pollen grains from the anther to the female organ by wind or animals. Pollen are good biostratigraphic and paleoenvironmental tools. This is the pollen grain. The next one is the morphology of embryophytic spores. The embryophytic spores are divided to three types: terylate, monolate, alate. Terylate is a spore with three apertures divided to three apertures. Monolate, a spore with a single aperture. Alate, a spore without a aperture. And this is the surface of sculpture of spores types. The next one is the spore description. And the spore description is how to identify the spores of five types: spore type, type of view, amp, lasure, and sculptures. The next one is the morphology of pollen grains. Pollen grains is polarity. Polarity means polar. The polar axis of the straight line between the distal and proximal poles of a pollen grain or a spore. The pollen grain can be therefore be isopolar, heteropolar, subsisopolar. The pollen grains identify to the previous one is polarity and next one is symmetry. The symmetry as spores and pollen are either symmetric or asymmetric. The symmetric ones can be A is radially symmetric and bilaterally symmetric. And the next one is the pollen grains of shape. The shape of pollen grains and spores can be three types. The first one is prolate, the second one is oblate, the third one is spheroidal. The next one is aperture. The aperture is the specialized thin region of spore or pollen wall that is generally different in ornamentation and in structure. The aperture can be in the form of pori or colpi. Grains with pori are called porate. Grains with colpi are called colpate. Grains with combined of two, colpus and porus, are called colporate. That's all. And these are the previous things of pollen morphology. Thank you.